Thank you for joining us at the cutting edge of cardiovascular sciences. I'm your host, John Cooke. I'm professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist Research Institute, and I'm joined by Dr. Lai Li, who's an assistant professor in the Department of Cardiovascular Sciences. And together we're hosting our speaker, Dr. Shahin Rafii. Dr. Rafii is the Arthur B. Pelfer Professor in Genetic Medicine and in Reproductive Medicine at the Weill Cornell Medical College. He received his undergraduate training at Cornell University and his MD from Albert Einstein. His laboratory has done some spectacular work in stem cell biology and angiogenesis that we're going to be hearing about a little bit about today. He is going to be talking to us about angiocrine factors in organ regeneration and in tumor growth. Looking forward to this presentation today, Shahin.
Great, thank you. Thanks for the invitation. I wanted to thank Dr. Lai and Dr. Cooke for inviting me to participate in this very prestigious DEBAKEY Symposium.
You've done some remarkable work over the years in angiogenesis, and we were talking just before the show about the interest that each of us have in translating our fundamental insights towards something that's useful. I think it's all of us that are MDs in basic science have that hope that someday our ideas might be translated into something useful, and you've made some real progress in that regard, haven't you, with a company that you recently started. Tell us about the company that you started and the clinical trials that are currently underway.
Yes, so as you know, the field of vascular biology or angiogenesis field in general was at 2 decades ago very important for health and also basic science studies. At that time, the whole idea about blocking angiogenesis was the hallmark of many meetings, and pioneer in that field, Judah Folkman and many others, were focusing on killing endothelial cell, blocking endothelial cell function, interfering with vascular function, with the idea that depriving tumors from blood supply and nutrients will result in regression of the tumor. So for several decades, antibodies to different angiogenic factors went to clinical trial. There was benefit, but unfortunately, the benefits for anti-angiogenesis therapy for tumors were marginal, even though for colon cancer there was definite benefit. Pioneers like Dr. Napoleon Ferrara showed that blocking VEGF for colon cancer can improve survival, but for many tumors it was not effective. And while we are still many companies, many academic centers, they are trying to find out the mechanism and improve targeting of endothelial cells, my group was focusing on the other side of the coin. Can we use endothelial cells and vascular biology for promoting organ regeneration rather than blocking them to block the tumor growth? And as you know, currently for regenerative medicine, there are very few pharmaceutical drugs or cellular therapeutics approaches that can penetrate the patient with lung damage, heart damage, brain damage, diabetes. So when I started my career at that time, we became with the idea that each organ has a different type of endothelial cell, different vasculature. And we found out that the vasculature are not just inert plumbing. They produce a lot of growth factors, which we call angiocrine factors, that in an organ-specific manner promote regeneration and repair after each organ is damaged. That was a pretty outside the box thinking, because if the endothelial cell can do that, can be capitalized on their potential for therapeutic purposes. So while mesenchymal stem cell, mesenchymal stromal cell have been in clinic for years, there was no endothelial expansion strategy that could have been hosted by a company to go to clinical trial. So about 10 years ago, it became with the idea of maybe we can capitalize on the pro-regenerative function of endothelial cell for organ regeneration. I developed a technology to be able to grow human endothelial cell in GMP, good medical practice setting, where we can expand significant amount endothelial cell that is appealing to FDA with large margin of safety for transplantation for organ regeneration. And at that time, in academic setting, it's very difficult to bring the potential of endothelial cell to clinic. So at that time, I was lucky enough to get attraction of some angel investors, some other biopharmaceutical, and we started a company called Angiocrine Biosciences, where originally started at Cornell, but then they moved to California to benefit from California Institute of Regenerative Medicine funding for biopharmaceutical. And one of the goals of Angiocrine Biosciences to produce in a scalable fashion human endothelial cell, which is currently allogeneic, and to be infused for treatment of wide variety of disorders. So in a phase 1 clinical trial that was just completed almost a year ago, Angiocrine Bioscience scientists showed that infusion of human endothelial cells, which were derived from originally human umbilical vein, so they represent generic endothelium, into patients who are receiving high dose of chemotherapy and bone marrow transplant, and as such their vasculature are damaged by high doses of chemotherapy, whether infusion of endothelial cell could mitigate the damage to those organs. I just have to mention, disclose, I'm a co-founder of this Angiocrine Biosciences, but I'm not paid in any sort of fashion, so what I'm disclosing you is what is in public domain. So in that phase 1 that was also announced to the public domain, it was shown that there is a trend upon intravenous endothelial infusion, mitigation of gastrointestinal toxicity in those patients who are getting high-dose chemotherapy and bone marrow transplant. There was a trend also to hematopoietic recovery, but most of the benefit from that phase 1, phase 1b was benefit of those gastric toxicity. Because of this positive signal, FDA has given a green light to Angiocrine Bioscience to move to a phase 3 multi-center double-blind clinical trial when the efficacy of the endothelial infusion, this is the product called AB, Angiocrine Bioscience 205, for mitigation of high-dose chemotherapy damage to multi-organ to the vasculature.
That's spectacular. We have a very busy clinical trial practice here, and would love to participate in that. That is very cool. This endocrine, this use of endothelial cells for therapy. Wonderful. Well, the congratulations on getting that far, bringing your ideas to the clinic, and I think we'd like to hear more about the basic science that you're doing that supports the work that Angiocrine is doing, and what you plan to do in the future. So maybe this would be a good time to share your screen and show us your most recent basic science on the role of the endothelium on maintaining organogenesis and homeostasis. We saw it there for a moment.
Yes, I want to share from this other screen. It's nicer here. You see it now?
Beautiful.
Okay, great. All right. So the topic that I'm going to show you is some of our recent, which actually, because you were so interested in biopharmaceutical, this new data I'm going to show you, which is a follow-up of the previous studies, is a topic of another pharmaceutical vascular biology company we want to start, that we're calling it Free Solo Therapeutics. And I'm going to tell you why we have called Free Solo Therapeutics. So as you mentioned, John, and I mentioned, and Dr. Lai, you know, we are desperate to bring the potential of stem cell physiology, and which I know the Methodist hospital is heavily involved with that, and your groups are involved with that, to the clinic. And unfortunately, so far, because of the poor stem cell lead or poor vascularization, the potential of therapeutics for regenerative medicine has not been realized. And in fact, there's no approved FDA drug for regenerative medicine except bone marrow transplant. Question is why? Why, despite numerous FDA approval for CAR T-cell therapy, immunotherapy, for cancer therapeutics, regenerative medicine is falling behind? Maybe the answer is because we are thinking too much to focus on drugs, but I don't think drugs are going to cure regenerative medicine problem. We need cellular therapeutics. And one of the solutions would be endothelial cells. As I mentioned, can be infused or rebuild organs. But the problem of growing organ-specific tissues is that we have come to realize endothelial cells within each organ are completely different and they produce different angiocrine factor. For example, in the liver, they produce Wnt, angiopoietin. In the lungs, MMP14, EGFs. In the cardiac, neuroregulin, agrin. Brain, BDNF, and every other organ produce different organs. So we believe there are 240 different endothelial cells within human body in mouse. And I would propose today that if we want to successfully regenerate organs, make replacement organ, vascularize, we cannot just put generic endothelium. We need to engineer organ-specific endothelium that can produce the right angiocrine factor, right adhesion molecules, right growth factors. How can we do that? So I just give you an example. We believe that transcription factor play a major role. I give you this example here. This was just accepted that it's going to come in cell stem cell. The idea that how liver endothelial cells, for example, the sinusoidal endothelium get specified. You know, in the liver, for example, these sinusoid that link portal vein to central vein are very unique. They're fenestrated. They regulate many aspects of hepatocyte co-zonation. And we report in this paper that the BMP9 produced by hepatocyte through activation of a transcription factor called CMAF, and in collaboration with GATA4, specify generic endothelium to become sinusoidal zonated liver endothelium. And this is a very important finding to my judgment, because not only it opens up our eyes that vasculature, by having this induction of these angiocrine factors and these transcription factors, can acquire this uniqueness, but also maybe we can use it for therapeutic purposes. In fact, it is believed at the moment that during development, ETV2 fly-ERG give rise to the generic endothelium, then extravascular cues like biomechanical forces, oxygen tension, substrate elasticity, microbiome, listening to news, any stress, turns on this transcription factor in an organ-specific manner that we get blood-brain barrier. We published recently, for example, TBX3 makes glomerular endothelium, and here CMAF. And in any of these publications, we knock this transcription factor selectively on endothelial cell, and they lose their specification. So it seems that this concept of organ-specific induction of transcription factor, which follow-ups of John and Dr. Lai, your own finding of trans-inflammation regulating this resilience of endothelium, seems to play a major role in conferring, getting the extravascular cue signal, turning into an organ-specific manner. But how can we study this? How can we generate a generic endothelium to figure out how external cues turns on in transcription factor? Like any other group, we started to look at iPS, induced pluripotent stem cell, but these cells are extremely tricky. I want to highlight this thing because this paper that was published by Littman and Schuster years ago, and they claim that they have made from iPS, induced pluripotent cell, blood-bearing barrier endothelium. They look like endothelium, they had tight junction, but Rafii and Tyler in our lab showed that they are not endothelium. They are EBCAM positive epithelial cells. And there is a lot of arguments going back and forth. Brain endothelium or any endothelium do not express EBCAM. And we published about this, but this is, and I think the reason this happened is because iPS-derived endothelium are very tricky. They are very unstable. They cannot be passage. They can be mistaken as shown here further. So what are other possibilities that where we can generate in generic endothelium? Can we make an mature adult human endothelium be reprogrammed to a durable, perfusible, adaptable endothelium to study organospecific? So this paper that was published 2 years ago, Brissot, a grad student at that time, was leading it, hypothesized that this pioneer transcription factor that is turned on during embryonic development and specify endothelium, but it turned off mid-gestation. If we turn it on again transiently in adult endothelium, maybe we can reprogram adult endothelium to an adaptable tumorigenic endothelium. And we call this reprogram vascular endothelium. Basically, we generate a endothelial cell that are now have no memory of organotypic. They are resilient, they are adaptable, they are generic endothelium. These are very different from iPS endothelium. iPS, you take an adult cell, you go all the way to embryonic development. This is taking a child, a student, bring him all to kindergarten. Here, with ETV2, we bring a professional, bring him back to college. Now this endothelial cell can get different courses, become engineer. They can get another course, become a lawyer. They can get another course and become scientists. We showed that this endothelial cell can self-assemble in this huge, without any synthetic barrier, with defined metrics such as laminin and actin collagen, no serum, in this large network of vascular net, which doesn't require any scaffold. They have lumens, they are polarized, and more importantly, they can be decorated by pericytes. You can inject them subcutaneously in mice. They can anastomose to mouse circulation for up to 10 months. Whoever can show me an iPS-derived endothelium can go beyond 2 weeks, I submit that this. But this is the power of human endothelial cells that are programmed. 10 months, no toxicity, no problem. More importantly, as I mentioned, they are hierarchy, they don't require scaffold, I'll show you a movie of this. You can get this RVEX as shown here in green. You put them on matrix in 3 dimension without any synthetic barrier. Defined matrix, it can be fibrin, laminin, and actin collagen. They form this beautiful 3-dimensional network. They can put him in any large microfluidic devices as shown here, and they self-assemble to these 3-dimensional vessels. And most importantly, this can carry human blood. They can carry immune cells. They can carry anything you want. You can study, you can put them with organoid, tumor organoid, normal organoid, push T cell, B cell, dendritic cell. You can push chemotherapy, you can push drugs, you can push anything that you want. You can change the metabolism. So this allows you to really study that. And because you can put in large microfluidic devices, we can make this humongous, large, without any constraints for study their biology. You can do elegant imaging on them. We can, for example, here red blood cells are shearing through at a certain dynes per centimeter square on endothelial cell. And when we published this paper, nobody believed we can get this extracellular magic. But just to give you a break here, it's like an Avatar movie. We are now going to travel inside these vessels. We are going inside these vessels. As you see here, they are branching, they are sprouting, they have nice junction. You can go in three dimension up and down. There is junction right there as you pass about. And as such, they're really three-dimensional blood vessel that, to my knowledge, no other technology that allows to do that in such a physiological manner, different sizes, different shape, different scope, different capacity you can see here. As I mentioned, can push blood. You can look at them after chemotherapy, for example. You can see they're insufficient, or you can, for example, in COVID, you see they become thrombotic. We can measure factor dimension, lacunarity, vessel density, branching, angiogenic index, and vascular health. We can give all sorts of different things. Now, there is a competition. I wouldn't say competition, but there is like Emulate and other groups have this organochip model that is well received. The problem with this model is that there is a synthetic porous membrane that separates epithelial cell from endothelial cell. We just talked about social distancing by Dr. Cooke. This is exactly the same. There is vascular distancing. The pores between epithelial and endothelial cells are very small here. Endothelial cells produce many contact membranes, KIT ligand, Notch ligand, HGF, thrombomodulin that is the cause of COVID, EPCR. None of these are exposed in this Emulate system or any organ chip model on endothelium. With our system, stem cell and organoids are in direct cellular contact with the endothelial cell. If you see here, this, for example, this red is one hematopoietic stem cell, attaches to endothelium and expand and self-renew. So I think our system is one step ahead of this system that requires this synthetic biomembrane to cause social distancing between the cells. And as an example, because of this freedom to operate, these RVEX can adapt to organoids. Here is human pancreatic islets in the lower panel, put on with RVEX in 3D in microfluidic devices in any type of matrix. We call them organ on vascular net or pancreatic islet on vascular net. And as you see here, if you put islets with this endothelial cell, this endothelial cell immediately vascularize these islets and they penetrate deep into this islet in 3D. You can push blood, you can push immune cells, direct cellular contact in 3D. You cannot do with other organ chip model. And as such, you can study many of the physiology. And by the way, you can do this with any type of organoid that I'm going to show you. Lung organoid, brain organoid, you can do with any type. It's going here. I just want to show you here that these islets, if you show in here, these islets are, insulin is blue, EpCAM is red. They are completely vascularized by this RVEX. I think you can see how they penetrate deep into this islet. As you see here, there is direct cellular contact between endothelium, as you see in physiology, with this islet. So this is the key to success, to generate tubulogenic organoids for transplantation. Let the endothelial cell learn to become like islet endothelium because of direct cellular contact, and also for mechanistic study. In fact, if you get large microfluidic device, you push glucose. At the outlet, you can get insulin, lactate, and glucose. So this is physiological vascularization of human islets. Since I have a short presentation, we have transplanted this in mice. We have cured mice from diabetes, and we hope that we have gotten funding from JDRF and many other avenues. We're hoping within the next years, this vascularized islet can be injected sub-Q, not into the liver. That is done with the Edmonton protocol.
Shahin, may I ask you a question?
Please, go ahead, please.
The vascular net, this is really fascinating. What's the scaffold for the vascular net that you have generated?
Excellent. So, as I mentioned, we don't have any biosynthetic membrane here. When we put this endothelial cell in this large microfluidic device, John, we use a matrix called laminin and tactin collagen. We don't use matrigel. Matrigel and serum are poison. Don't use them in your lab, should not be used. Matrigel come from human tumors in mice. FDA would never allow you to use matrigel. Serum, if you use serum, none of these vessels form. So the scaffold for these can be fibrin, laminin and tactin collagen. We're working with hyaluronic acid. So I think for each organ, we're going to have custom make matrix.
That makes
no
sense.
synthetic biomembrane here.
Sure, that makes sense. So the vascular net is composed of extracellular proteins, extracellular matrix proteins that you'd expect to see in a blood vessel.
Correct.
Beautiful. And then how do you actually fabricate it? I just was curious to how that was done.
Yes. So if you see here, this, like if we have this inlet and outlet here, we fabricate these large microfluidic devices that carry about 26 microliters here, and because the reason you're asking this question, because it's very to conceive that this endothelial cells self-assemble and pattern in this large microfluidic device without any synthetic biomembrane. That's the powerful of ETV2, that transcription factor I mentioned to you. Somehow this transcription factor generates the program for plexus for the primitive plexus that's formed in human during development. So we don't need any synthetic biomembrane. We don't need any scaffold per se. They automatically self-assemble into these large microfluidic devices.
Beautiful, thank you.
In the presence of the matrix, of course.
Wonderful, thank you, Shahin.
No problem. So infusion of glucose result within the outlet, and you can study any type of metabolism here. And because we have so many endothelial cells, you can take your endothelium, do seahorse. You can study oxidative phosphorylation. You can do trans-inflammation, which is John is famous for that. So this is another rendering where we can put iPS-derived cardiac myocytes. This is for you guys because you work with the heart. We can vascularize with RVEC. In here is in fibrin as a scaffold, John. So there is no matrix, there is no scaffold per se. This endothelial cell just vascularize this myocardial cell. And as you see, I can push blood. They're contracting. Oxygen goes through. This is my blood, by the way. And you see they're just contracting. So basically what we can do here is vascularize any tissue. And I would argue, you see, I can go fast, I can go slow. I can ready to transplant this in ischemic myocardium. So, and they immediately anastomose to myocardial tissue. This is, for example, colon organoid, human colon organoid. We put it with these vessels, no scaffold, no synthetic biomembrane. They immediately vascularize this organoid. Now you can study interaction with endothelium with the organoid, with any system. And these are in very, very large system. You see, and then each of these organoids get connected as well. In fact, you asked me about this. So all the data I showed you was in 15 microliter volume. We can go to 26 microliter volume. I can even go to the 5 centimeter microfluidic device that can accommodate 200 microliter of matrix. Here we have in the 15 microliters maybe 50,000 endothelium, 26 microliters, 100,000. Here we have 4 million endothelium. And because of that, I can do flow cytometry. I can do metabolic analysis. I can do molecular and proteomic profiling as well. So we have done brain organoids with Rafii list for brain. We have done intestinal, we're doing lung organoid. We can vascularize any decellularized scaffold. One of the biggest low-hanging fruit in regenerative medicine, when Doris Taylor in Texas Heart make cardiac decellularization, her major nightmare is to vascularize those small vessels. We can vascularize for her. So the idea of moving forward, hopefully if this free solo therapeutic company start, the idea is to make replacement organ. This is different from Angicom Bioscience, when we infuse endothelial cell. Here we put our organ on vascular net model at work. We can get organ-specific stem cell, organoids, put it in our vessels, transplant subQ for diabetes, injecting the heart, injecting the brain. Sky is the limit what we can do with vascularized organoids. So if you have any question, I'm going to now shift for a few more minutes to the cancer biology. But if you have any question related to normal physiology, I'm open to any question that you may have. If not, maybe I
think,
proceed.
why don't you proceed? That was fascinating, and
Okay.
I think in the discussion period we can ask a little bit more about your vascular net and its applications. But
Yes.
yeah, please, Shahin, please proceed with the maladaptation of the vascular niche in tumorigenesis.
Thank you so much. So far I have shown you that endothelial cells are adaptable, and if you adapt nicely, you cause organ degeneration. But if you have chronic injury, oncogene activation, IGF, jagged, ETS2, you can get bad angiocrine factor and you get maladaptive repair. In fact, my group and another group have published that the reason CAR T cells do not work for solid tumors is because endothelial cells don't let CAR T cell go into solid tumors. Chemotherapy resistant is because so-called tumor stem cells are fed by bad endothelial cells. Can we model this with our organ on vascular net? Absolutely. We can put tumor organoids with our RVEX parasite 3D matrix, no scaffold joint, no synthetic biomembrane. You put this tumor with endothelial cell, they get vascular, exactly what I showed you with colon organoid. I can put up to 50 colon organoid, and each well consists of 2,600 tumoroids, 200 RVEX, 20,000 parasite. You can do single cell RNA sequencing, ATAC-seq, metabolomics, anything you want. I give an example of this because I think time is limited, what we can do with that. What I'm going to show you here, which was done collaboration with Dr. Laura Martin in New York Stem Cell Foundation. Here we vascularized, the red is ovarian tumors that are HER2 positive. Monsojin and Yamping from our group made CAR T cells that are blue here against HER2. And we infuse them in these microfluidic devices. You see this blue CAR T cell transmigrated and they targeted the tumor. The yellow is when the membrane of these ovarian cancers get targeted, dye goes in and they become blue. You are seeing for the first time how CAR T cell kill tumors. To my knowledge, no other group have been able to really show how CAR T cell kill the tumor. Red is an ovarian tumor. CAR T cell to HER2 is persistent, identify a tumor, is injecting granzyme, and you see that yellow is a death. What we saw is amazing. You see suddenly the other tumors become yellow. Now we can, in this rendering, there are 50 tumors. These CAR T cells were only killed, for example, you see this yellow was targeted. They only killed 70% of the tumors. We don't understand why the other tumors were not killed, but this model can make us understand why CAR T cells don't target solid tumor. How can I change the endothelial cell so they can start to target the tumors? I can give a whole lecture about this, and what is amazing is that the endothelial cell in this system also becomes tumor endothelial cell. They maladapt and they become a nightmare. And I think by this system we already have found some clues how to change endothelial cells to maybe normalize them, as Rakesh would say, or how to change them so that the CAR T cell can't do tumor. But one of the most important data that I'm going to show you here is in this slide. I showed you that in addition to the capacity of this RVEX, reprogrammed vascular endothelial cell, to be so tubulogenic to be so acrobatic, to self-assemble in 3 dimension, so effective to anastomose to the mouth circulation, which is what else they do, they adapt. I can make a college education from Dr. Lai. Imagine Dr. Lai, who's a top scientist, says to Dr. Cooke, I don't want to work anymore. I want to become a lawyer. Now you can give him Oct 4, bring him all the way back to when she was a child, and a lot of things can go wrong, right? Or I can give ETV2 to her, bring her to college. Now in college, she can start to take courses to become maybe a lawyer. Arvex, exactly like that. For some reason, their chromatin is very adaptable. And as example, if you put Arvex with colon organoid for 7 days in direct cellular contact, and then let them co-educate each other, take them out, do single cell analysis. The blue is the educated Arvex. The red was the control Arvex. You see here now, new endothelial cells have shown up in single cell that have a new signature. Now they express PLLV, TFF3, FAB1. If you look at Peter Carmine's listen data, these endothelial cells are approaching to become intestinal endothelium. The chromatin got educated. Now, as I mentioned, we want to study what metabolism changed here, many other things. By the same token, if you put this Arvex with colon tumor organoids for 7 days, they become, just look it up. This red is the endothelial cells, Arvex. Look, they all change to these blue cells. And if you look at those blue cells, they acquire tumor vessel signature. Years ago, me, David Lai, and Robert Benezor showed that IT1 is upregulating tumor endothelium. Look here, IT1 was induced in Arvex. So basically, I was able to produce tumor endothelium. So
Shahin,
ETV2,
may we ask you a question?
Hi, Dr.
absolutely,
Rafii.
please.
Can I have a question? So, based on the slides you just right now, you're showing us that you see the refs can acquire different signal, different profile after you co-culture with other tissue organoids, right?
Yes.
But do you expect that? Do you see that those co-cultured refs express other signatures of other kind of endothelial cells? Because to me, I feel the ETV2 overexpression kind of bring the endothelial cells back to a more progenitor stage. So I wonder if they also express some other signature of other endothelial cell markers.
Very good question. Very good question. So I forgot to tell you, which is very bad. We only express ETV2 for 1 week. For 1 week. Minimum 1 week. 1 week is enough to remodel the chromatin because it's a pioneer factor. And we have looked at the single cell analysis to answer your question. For example, with this Arvex with the tumor, they really become tumor endothelium, not anything else. Or here, when they become the intestinal endothelium, you see they become very close to become intestinal endothelium, and they are very focused. Now the question is how long this lasts. We haven't studied that, but we believe for a few weeks, as long as they are with the colon organoid, they stay like colon endothelium, but they don't acquire cardiac endothelium or brain endothelium, if that's your question. They really stay focused.
And also have one more question. So when you overexpress the ETV2, do you see batch variation? Because if I look at the single cell clustering picture on the top two, I feel there are a lot of difference.
Correct. Absolutely. Yes. There are some differences that because of the microfluidic devices, but if you look at the signature of those reds that you show in there, that's a very good point. They are probably 90% very close, and that's the way we will process them from the tissues. But The reds are usually very, very similar, and that's on the paper. But you are absolutely right. When we did this study, we had to use 10 different concentration of ETV2 lentivirus. And we found out there is Goldilocks. Goldilocks means perfect dose of ETV2 to get this. If you use high dose ETV2, cells die. If you use ETV2, nothing happens. So to get a batch of ETV2, we get maybe human umbilical vein. By the way, this works for any adult endothelium. You can get brain endothelium, you can get heart endothelium, put ETV2, they get reprogrammed. And
Could I
we found
make
out that
a comment on that, Shahin?
Yes.
You talked about a Goldilocks zone and in the delivery of the lentiviral ETV2. You also mentioned that ETV2 is a pioneer factor, and I don't think there are any pioneer factors, actually. I think that to change the phenotype of a cell, obviously you need DNA accessibility, and you need transcriptional direction. And the idea of a pioneer factor is that the pioneer factors provide both. They provide DNA accessibility. They're pioneer factors. They can get into the chromatin, through the chromatin, and have an effect on the gene, provide transcriptional direction. But you're delivering the ETV2 with a lentiviral construct, which is activating inflammatory signaling that we've shown is absolutely critical in DNA accessibility. So I don't think there's any pioneer factors. I think the confusion has been when you deliver these transcriptional factors, you're delivering them with vectors, vehicles that induce inflammatory signaling, the transflammation. Transflammation is what you brought up a few times, is two things. It's inflammatory signaling that provides for DNA accessibility, and then you need transcriptional direction. So you need both. And I just thought I'd make that comment when you raised the issue of pioneer factor.
Yes, that's perfect. How about if I tell you that I can get the same result by using modified RNA of ETV2?
Modified what?
RNA.
Modified RNA that express ETV2.
Yeah, I believe it. But even modified mRNA can induce slight inflammatory signaling.
Yes. And as I mentioned, we are following your beautiful publication years ago about transinflammation. And in fact, what we are doing right now, exactly what you're saying. We're blocking NF-kappa B, AAP2, following exactly what you're saying. Is it ETV2 is a pioneer, or it activates this inflammation to open up the chromatin? In fact, I knew you're going to ask that question, and we have done those studies. So we did ataxic analysis on after ETV2 transiently, and as you see here, exactly what you said. What ETV2 does just opens up in a vascular specific manner the sites in enhancer element that was shut down in an organ-specific manner. For example, in liver endothelium, response to physical stimuli is shut down, but ETV2 opens it up. And the mechanism, when you do H3K27 acetylation, H3K2 trimethyl, you see that what ETV2 does, exactly what you said, increases the enhancer element of wide variety of silence generic endothelial marker, such as, for example, as I mentioned, here you can see DLL4, collagen, many things that are shut down in an organ-specific manner. It doesn't get turned on, it becomes poised. It becomes poised. Look at K4 trimethyl here, K27. So basically they are poised now. So the methylation is not resilient. Now you give it external stimuli with the tumor, boom, it won't get turned on. Exactly what you said. So I think what happened, ETV2 reactivates sensor for mechanical and remodeling gene in adult endothelium. You are right, it may not be a pioneer factor, it just be this cross-inflammation, and ETV2 somehow enabling access allows this resilience to take place. But it's only ETV2 does that. If you do fly ERG, it doesn't work. There is something about ETV2 DNA bonding domain that does that. So let me go to the last part of the story just quickly. Now, I mentioned in the beginning, I'm going to go through this fast because we already discussed that, can be used endothelial cell for therapeutics. This was an angiocrine bioscience that John asked me a question and I mentioned it. So the idea was, if you have organ-specific damage, if I transplant endothelial cell intravenously, can they go and graft into damaged tissue such as lung, produce the right angiocrine factor, and repair the damage? And I can do for any organ. And just an example, this was published by Augustin Choi Ardeen and Fernando Martinez. And in this study that was spearheaded by Alex Racanelli, who's an attending physician at Weill Cornell, we gave elastase to mice. Elastase, when you give to the mice through the aerosol in the lung, you get lung damage and the lung partially regenerates. But it regenerates fibrosis, so this is a model of COPD or emphysema. But if you infuse 500,000 of this endothelial cell we have engineered with this gene called E4F1, that is a product of angiocrine biosciences, as you see here, the mean cold volume goes up. Not only that, inspiratory capacity and the lung resistance elastase goes up. Improve. Therefore, these endothelial cells are homing to the lungs. They're producing angiocrine factor. And I have published a series of Nature Cell papers that endothelial cell can be anti-fibrotic by activating CXCR7 and blocking TGA beta. And we showed here that the infusion of endothelial cell can reverse even elastase-induced lung damage. We're working right now with our company and Cornell to maybe we can even take your lead, come to you to use endothelial infusion for patients with a very severe COVID damage in the lungs. Because, trauma modulating down, there's microangiopathy. Does endothelial infusion of a healthy endothelium, shown here, can mitigate microangiopathy in COVID lung patient, also promote regeneration. So infusion of generic endothelium mitigate elastase-induced pulmonary injury. So I mentioned this endocrine bioscience, the AB205, they call it universal E-cell. This came from HUVEC, they're allogeneic. This was the first US multi-center open label. They escalated the dose to determine the safety and efficacy. 31, I think this is up to 42 now, lymphoma patient were going curative high-dose chemotherapy. They had multiple damage in the GI tract, bone marrow, mucositis. First, what we saw here, this was the first endothelial infusion ever to any human being worldwide. I think I'm very excited about that because we infused the first endothelial cell without any toxicity. And we showed for the first time endothelial infusion is safe in 42 patients. So I think that was one advance. And second advance was that even in the phase trial, 90% decrease in gastrointestinal toxicity from grade 3 to grade 1, 70% decrease in febrile neutropenic fever, and trend toward rapid platelet in 20 patients. As I mentioned, based on this data, they have gotten rapid regenerative medicine status. And in phase 1 clinical trial, they're going to do 120 patient, placebo control, multi-center, and hopefully we can come to you guys as well. So what I've shown you that we may one day don't need any mice anymore. I can get our organ on vascular net, put bone marrow in one microfluidic device, tumor in one microfluidic device, lung in one microfluidic device, liver, islet, kidney, serially connect these vascularized organoids together, and then I can push chemo, any drugs you want, and check the toxicity. We call this human on vascular net. I think we're testing this on right now. We can screen for cancer targeting, personalized screening. Every time we can bring a patient biopsy sample, I have fat tissue, endothelium, and I can do personalized medicine for that. So, and this is our goal, eventually have this vascularized cardiac endothelium to inject them into ischemic myocardium. I mean, this is fantasy for us, but hopefully with funding and collaboration, we can inject them to a damaged myocardium allow this endothelial cell produce the angiocan factor and repair the damage. So in summary, I have shown you we have entered an era of translational vascular medicine beyond angiogenesis. We can make tissue-specific vasculature, hopefully with ETB2 and many other transcription factors. We can make perfusable, adaptable endothelium to uncover inter and intra-organ vascular heterogeneity, enable immune screening and cause T cell. And eventually, I think in few years, by the way, any cells you buy from pharmaceuticals, Lonza, you order lung endothelium for $700. Don't waste your money. Those endothelial cells from lung lose their specification as soon as you put in tissue culture 20% oxygen serum. I believe in next few years, by making this transcription factor, we can make kidney endothelium, brain endothelium, liver endothelium. You don't need to buy anymore. So I think this is my vision of where vascular biology goes. I mentioned a lot of people have played in there. I mentioned their name throughout. It takes a army to work with vasculature. It's very difficult. I mentioned many names. And also I want to mention that thanks to Dean Augustine Choi, recently we have inaugurated Hartman Institute for Therapeutic Organ Regeneration. This is follow-up of Ansary Stem Cell Institute, is complementary to that. And the idea would be to take stem cell for Ansary Stem Cell Institute, put them in Hartman Institute, and make replacement organ. We have made with Todd Evans electrophysiology lab. The only thing we are missing, GMP and large animal models, pig models. So anything we can collaborate, I'll be more than happy to do that. In that note, I think I stop, and sorry I went too long, maybe I was too excited, but if you have any questions.
Shahin, that was just marvelous, and I want to tell you, I am proud of you. That was so spectacular. You've done such a great job going all the way from the basic science to clinical application. I mean, what a marvelous thing the vascular net is for understanding organogenesis and how the endothelial cells modulate organ homeostasis. What a wonderful thing it is for studying tumor and understanding tumor therapies like the CAR T cells you showed us. And the endothelial infusions, I'm excited about that. I tell you what, we want to work with you. We have a new stem cell center here. I would love to work with you. We're very translational. Love to get your ideas into the clinical trials. Let's do that. And you know, maybe we can even help with our Center for RNA Therapeutics, because we can make clinical grade RNA and deliver it with clinical grade lipid nanoparticles that could provide the transcriptional factors to give you the cells that you need for a particular application.
That would be awesome, because I think the epigenetic memory from this transcription factor, even with modified RNA, that's a brilliant idea. I think we need to have, thanks to you inviting me here, every month we should have a symposium when your groups present to us, our group present, to set up these complementary interactions.
Let's do it.
And I think it would be tremendous, put the forces, both institutions together.
That'd be spectacular. There's a lot more to talk about. We don't have that much time, but I want to give Dr. Lai a chance to ask a few questions.
Yeah, sure. That was a very good talk. I get inspired by your talk, actually. So I have a couple questions. The first question is, I think your vascular net is kind of mimic. It's an in vitro system to mimic the in vivo world, right? But I just want to ask you, what do you expect if we want to see, especially what does, especially under some specific disease condition, so how would your system work?
That's brilliant. So the system is so resilient. For example, AVM, arterial vascular malformation. Recently, Dr. Chris Hughes showed beautiful study with his microvascular system. So I can put mutations, like type 2 mutation that cause AVM. I can put it here, and I can make AVM, be working on that like Chris is doing, and then screen for drugs. Exactly what you hit it right on the mark. I can put with CRISPR-Cas any mutation you want, or any drugs that can cause chemotherapy mediated, mitomycin-induced mutation. Sky's the limit. So I just need to hire a postdoc and work with you guys. That's an excellent question. We can model any disease you want.
Yeah, I'm
Vascular
looking forward
disease
to hear your future work about this part. And I have another question is, because you were talking about the heterogeneity of the endothelial cells, so, but in the tumor field, there is different principle that showing the tumor cells has the ability for the endothelial cells to become stem cell or metastasis. They're either determined genetically, or they are like what you said, educated. So what do you think about the what to determine the endothelial cell heterogeneity?
Yes, that's a good question. As I mentioned, when you buy this endothelial cell from drug, Lonza, they're all. And by the way, many of those endothelial cells you buy are lymphatic endothelium. Actually, they have PROX1, and the company doesn't tell you. We're doing single cell analysis on them. If you look at those endothelial cells, they always have a baseline signature. Goes back to your epigenetic comment. You see a vascular epigenetic signature that is common to all these generic endothelium. I think that exactly what you put in embedded there, endothelial progenitor, endothelial stem cell, that baseline epigenetic is dictated by FLY1, ERG, and ETV2. I didn't have time. We have a paper on the review right now. We knocked out FLY-ERG in adult mice, endothelial cell disappear. So I think ETV2 is gone, FLY-ERG are the key to make that baseline epigenetic endothelium. Then microenvironment teaches those promoters of those regions to turn on MAFC, as shown here, TBX, GATA4, and they become specialized. So you're absolutely right. There is an epigenetic information to stay endothelium. But to become heterogeneous, I think you need an education to become specific.
Just along those lines, I really like your idea that whereas the endothelial cells are of an organ, are maintaining its homeostasis, organ homeostasis, the organ is also maintaining a certain endothelial cell identity. There's communication between the endothelial cells and the organ that makes them compatible. You also talked about the tumor microenvironment, and we know that the tumor microenvironment plays a role in the resistance of the tumor to CAR T-cell therapy and other therapies. Yes. What is the role of the tumor endothelium in resistance to the immune surveillance and CAR T-cell therapies? What is the endothelial cell work? What are endocrine factors are being produced that contribute to tumor resistance?
That's a beautiful question. And, we published 2 cancer cell papers, others, and I think your colleagues have done as well. When the tumors become too hypoxic and the tumor produce excessive amount of, for example, EGF or Notch ligand, endothelial cells lose their specificity, and transcription factor like ETS1, which is an oncogene, or ETS2 get induced on endothelium. Then suddenly you get a lot of Notch ligand going up. A lot of wind goes up. All of those, the tumor aberrantly corrupts the endothelium, corrupts and customizes for its own benefit. At the same time, we are seeing tumor tell the endothelial cell to shut off his immune activation, and in fact, it upregulates PD-L1 to prevent the stuff to come in. We see this over and over and over again, that PD-L1 and CTLA4 and many other things are activated on tumor endothelium. And
Very
that's because
interesting.
the tumor are not talking to them.
I'm sure with your vascular net, you're going to learn a lot more as well about
absolutely
how the
more we
endothelial
do
cells.
this co-education and we do single cell, we can answer your question even more in depth.
Absolutely. wonderful model, wonderful work, and a lot of hope for the future. We've been hearing Professor Shahin Rafii, the Arthur B. Pelfer Professor in Genetic Medicine of Weill Cornell Medical College, who just gave us a marvelous lecture on angiogenesis and endothelial cells and their role in modulating organ homeostasis and tumor resistance, and future work on endothelial infusions for therapy, and just a wonderful lecture. Thank you so much for that great basic science, and I look forward to collaborating with you in the future, Shahin. Thank you for joining us today on the show.
Us too. Thank you so much for the invitation. Thank you.